Welcome back to the course on fluorescence spectroscopy. Today we're going to begin our discussion of protein fluorescence. This will be at least in two parts, even possibly three parts. The first one will be about steady state measurements of fluorescence and what we've learned about protein structure from the steady state measurements. First off, proteins are exceedingly complex. They're probably more diverse than any other macromolecules. Maybe carbohydrates are more diverse or more complicated, but they probably don't have as many repeated units. If you look at some of these protein structures, the one on the right looks like there's two globules there. One on the right looks like a singular globular protein. You see regions of alpha helix in this one. See some alpha helix here. Looks like there's regions of beta sheets. We're not going to focus too much on those specific features. We're going to focus more on the amino acids that make up the protein fluorescence, in particular those that are most relevant to us. So those complex structures you saw in the previous slide are made up of only 20 amino acids. Now that's a lot of building blocks. In DNA you have 4, maybe 5 building blocks, depending on how you count them. So all the diversities in the sequence. In proteins, the structure is not only a result of sequence, but as a result of the side chains of the amino acids. So you can see the side chains here. They all have a carboxyl group and amino group for hooking to one another and different side chains. I think many of you have probably seen this presentation of amino acids more frequently, where they're broken up into different classes. For instance, let's see, we have the amino acid with uncharged side chains. Here we have the amino acids with hydrophobic side chains. We have the amino acids with electrical charges. So you can also link them up in those directions and those ways, and they all come together in some way to form the structures we saw on the second slide. And I just got a couple more examples for you because they're just such interesting structures. This is a protein that accounts for some plant disease resistance, and GROL is a protein that helps proteins to fold after they're synthesized, or helps proteins refold after they're denatured. Used to be called heat shock proteins because if you gave microorganisms a heat shock, they would make a lot of GROL because they needed to renature a lot of proteins. Of course, this is a multi-subunit protein. This is a single subunit protein. So there's the examples. So the amino acids that we're going to be interested in out of all of these 20 are going to be these 3, and it's going to be the 3 aromatic amino acids. We have the phenylalanine, the tyrosine, and the tryptophan. Phenylalanine is just that, it's a phenyl group on the alanine amino acid. Then we have hydroxylated phenylalanine, which is tyrosine. And then we have tryptophan, which has the indole moiety, which is responsible for most of the emission that we see from many proteins. Over here, I put a space-filling image of a protein. It's not as space-filling as it could have been, but in spite of the ribbon diagrams that I showed you in the previous structures, the actual proteins, when they're folded up, are pretty much close packed. I remember years ago reading a paper by a famous scientist saying basically there is no free space within a folded protein. So, what can we learn about proteins from these three amino acids? Well, first let's look at their spectra. Uh, the other amino acids don't have any absorption out in this range, maybe some down in the 250 range, but the proteins that absorb 250 and longer are really only the three I just mentioned, the phenylalanine, the tyrosine, and the tryptophan. In terms of what we observe with proteins, uh, it's pretty hard to find a protein where you can see phenylalanine emission. I think I found a couple, but there aren't many. At a slightly longer wavelength, because it's hydroxylated, we have tyrosine, and tyrosine emission is occasionally seen. And then, of course, we have tryptophan, which is the dominant emitter from just about all proteins, has the longest wavelengths for absorption and emission. We'll have a slide later showing that if you excite around 300 nanometers, you really only excite tryptophans. And tryptophans are really unique because they are the most expensive amino acid to synthesize in terms of the metabolism of organisms. And they're the only amino acid that is coded by a single codon. And that will become important when we discuss a little later on the trip repressor protein. So these are the three that we're going to care mostly about. How do the spectral properties compare with the spectral properties of common fluorophores? So let's look at the common fluorophores first. They absorb and emit in the visible, maybe out to the infrared. They have high quantum yields, and they have enormous extinction coefficients on the order of 100,000 reciprocal molar, reciprocal centimeter. When we go over to the three amino acids that we're going to be looking at, we find out that they have rather puny spectral properties. Looking at the extinction coefficients first, they're much smaller. They can be on the order of 5,000 for tryptophan as opposed to 100,000 for 150,000 for CY5. So the proteins absorb a lot less light. Their quantum yields are generally lower than the commonly used fluorophores. And the excitation wavelengths are shorter. Now you put all these things together, and just a back of the envelope calculation would say that protein fluorescence should be much, much weaker from that of the extrinsic fluorophores. There's a couple of reasons why we don't always notice that if you're looking with a spectrophotometer. If you have a spectrophotometer that looks at that's capable of looking at UV proteins, it'll typically have a lamp, like a deuterium lamp, that gives you more light in the UV than out in the visible. You'll probably increase the gain on the photomultiplier tubes. I might even open up the slits. So they don't always appear this 1200-fold difference in practice. Another thing is for all of these common fluorophores, we have laser light sources or LED light sources. For these fluorophores, we've only recently gotten LED light sources, and maybe they're not as weak as they used to be, but the ones that I have in my lab at the moment are pretty weak. But when we move to time resolved measurements and single photon, time correlated single photon counting measurements, that weakness doesn't bother us too much because we have more light than we can use with the common fluorophores. So let's look at the spectral properties of each of the fluorophores. So what are the spectral properties of fluorescein, 3-letter code, TYR? If you really want to know what the spectral properties in a protein are, I prefer to work with the NADA derivatives. This one would be called NADA tyrosine for N-acetyl tyrosinamide. So it's a amide on this acetyl group here, amide here. And the reason these are sort of better model proteins because you don't have these free charges around the tyrosine when it's built into a protein. So it's common when you're studying the amino acids to use these NADA derivatives. Tyrosine has very simple properties. First of all, the excitation anisotropy spectrum is simple. We'll see in a moment how complicated it is for tryptophan. But compared to tryptophan, it's a simple anisotropy spectrum resulting mostly in one transition. Tyrosine is not significantly dependent on solvent. That's somewhat of an advantage because it takes it out of the complexity of comparing it with tryptophan, which is extremely sensitive to solvent polarity. But there is one complication with tyrosine that it's important to remember, and that property is that tyrosine can display an excited state reaction. You might remember in our earlier lectures we discussed an excited state reaction where the fluorophore reaches the excited state. I haven't put in all of the other levels there, and it has one decay time, then it relaxes, but the relaxation here is not a solvent relaxation. The relaxation is the loss of a proton, so it's emitting from a completely different state, and when that happens, you usually see the initial state go down and the subsequent state go up, and that's exactly what happens with acetate. If you put it in something that can pick up this proton here, these are two different solutions of acetate, the same pH, because there's so much more base around, meaning for the acetate here, you can pick up the proton. So what is the usual tyrosine emission becomes the tyrosinate emission, and the different spectrum here is really the true tyrosinate emission. The rest of it is a tryptophan emission. On this side, we have just the emission spectra at neutral pH and high pH normalized. So this is where the tyrosine and tyrosinate emission is observed if you are able to observe it. The pKa's, I've forgotten the exact number, but in the ground state, the pKa of tyrosines, I'll sort of make this up, are about 9. When you get to the excited state, the pKa drops to about 2.5, and so the moment you excite the tyrosine, it starts to lose a proton, but it can only lose a proton if there is some base around to accept the proton. So now we come to really the subject of today's lecture, which is tryptophan. And the tryptophan spectra and the anisotropy spectra are much more complicated. Let's just look at the anisotropy spectrum first here. This yellow line, you can see it's quite a mess. It doesn't look anywhere smooth. This is part of the tyrosine anisotropy spectrum that I put here. And this has been a headache for biophysicists for a long time. I think an unnecessarily large headache for biophysicists. But the current explanation is, which I believe is correct, is that tryptophan has two long wavelength absorbing states. One we'll call the 1LB state, and the other one the 1LA state. The one meaning singlet states. And this total absorption of tryptophan, which is structured now, because it's a combination of the absorption of the 1LB state and the absorption of the 1LA state. And this has caused no end of headaches. And just speaking from experience with grant proposals, too many proposals have been shot down because someone would have an issue with which state you're dealing with. But because the 1LA state is the lower energy one, the emission is just about always from the 1LA state. What do the transition moments look like in these molecules? In the tyrosine, it's pretty simple. We have a lower energy state along the long axis where the hydroxyl group is. So that's this state here. The quantum mechanical people might argue, well, it's a single state, it should have a single anisotropy. It turns out that's never true with mixed organic molecules in multiple states. So this is a simple anisotropy spectra. Here, the indole moiety is known to have two different electronic transitions which are perpendicular to one another. The 1LA state is this the 1LB state is this structured one which goes along, I would say, sort of the nonpolar part of the tryptophan residue. And we have the 1LB state, which involves the nitrogen, and that goes along the that axis going through the nitrogen. And that is probably what makes it a simpler state. As you can see, it's smoother here. And being lower energy, it absorbs at a somewhat longer wavelength. So this, if you're ever studying protein fluorescence and you're trying to do an anisotropy measurement, it is common practice to only use excitation wavelengths 295 nanometers or longer, so you can be assured that you are going into the lower energy 1LB state. These states start to get mixed up a little bit when you put the indole moiety into a solvent. So here we have the polar solvent, and it redshifts or decreases the energy of the 1LA state. If you look really close, or if you use your imagination, you can believe that the 1LB state is not sensitive to the polar solvent. The 1LA state has shifted to the red due to the polar solvent, and that's the reason why, if you're using a long wavelength excitation, you can excite exclusively into the 1LA state. Oh, this slide I put together just to emphasize where the absorption spectra are. So if you take 300 nanometer excitation, you can excite tryptophan without much difficulty. But you can see tyrosine and phenylalanine, they no longer have any absorption. So that's why it's safe to use the 300 nanometer excitation to excite the 1LL state only of the tryptophan. And that's the state that gives you the higher anisotropy. And since whenever you're doing an anisotropy measurement, you like to start with as high a value as possible, so you use the longer wavelength excitation. There has been confusion about whether a protein contains tyrosine or tryptophan, and several groups have reported tryptophan-like emission from proteins that were known not to have tryptophan. And I have some examples of that here. And as is true for all scientists, we argued about it for a while. But it turned out that these proteins that you would almost swear it looks like a tryptophan emission turned out to be tyrosinate emission, that is emission from the ionized tyrosine. Sometimes there's a mixed emission from unionized and ionized, and the same thing with this protein here. But this was just a mix-up which is not being debated any longer, but it was a concern in some of the earlier studies. So, you normally don't see much tyrosine emission from proteins. One of the reasons is it depends on where you excite it, of course. But another thing that happens is fret does occur from tyrosine to tryptophan. And the fret distances are not very different from the radii of these particular proteins, which are a human interferon. And this is a difficult slide to explain, but I'll do it anyway. So if you look at the monomer, and this would be exciting at a wavelength where you could see the tyrosine, you see a certain amount of tyrosine here, and then the dominant tryptophan emission. When you put this protein into the dimer state, which is where it's more happy, you see that the tryptophan fluorescence has doubled. That's great. These are, normalized to the amount of protein that's there. The tryptophan emission is doubled here, but the tyrosine emission hasn't gone up at all. So this was taken as proof that there is a resonance energy transfer between tyrosine and tryptophan. So that's another reason why you don't see much tyrosine emission from proteins. I think we could quantify here that since the tyrosine emission did not double when the protein size doubled, that half of its emission was lost by fret to the tryptophan. It was always hard to separate tyrosine and tryptophan, so I couldn't get really a perfect slide. But this was, I think, a very nice example where they took the co-protein from E. coli and they removed all the tyrosines and they put in tryptophan residues where they wanted to. So they chose different locations, some where the tryptophans were very close to one another, other times when they were further away from one another. And then they collected the emission spectra, typically both at a wavelength where you could see tyrosine absorption and a wavelength where you only saw tryptophan absorption. So to understand the slides, let's look at these one by one. Tyrosine 21, tryptophan 21 and tyrosine 24, they're over here in this protein construct. you can see they're close to one another, and in this construct, you see no tyrosine emission. These spectra here, the purple ones, are the different spectra as seen with excitation of both tyrosine and tryptophan, or excitation, which is tryptophan. So the close ones transfer to one another. Let's go down to the one that are furthest away, tryptophan 21, tyrosine 15. So here we have tryptophan 21. Here we have tyrosine 15. It's too long of a distance for energy transfer. So when you excite at short wavelengths, if you looked at just the emission spectra, you can see a little bit of the tyrosine emission on the blue side of the spectrum, and the purple is the different spectrum. So this is a proof that depending on the distance between the tyrosine and tryptophan, they will transfer energy. Another proof is given here, which are the excitation wavelengths. And I sort of like these. This is sort of a better proof to me. The excitation spectrum is where we vary the excitation spectrum, and we're just looking at the emission maxima. And what you can see with I think the 21 and the 24, which was the first one we looked at, is the excitation spectrum is higher than the other mutants that were made. So you can say that says pretty definitively that tyrosine was contributing to the tryptophan absorption. If we go to the pair that were furthest away, that excitation spectrum is lower because none of the excitation going into the tryptophan 39 is showing up in the tyrosine 15. So that's another way of showing energy transfer. In fact, that's the first way it was done. And there's another way of plotting this, which I decided not to show because it just gets confusing. Interesting thing about tryptophan is it's extremely sensitive to any contact with water. In order to solve the mystery of why tryptophan had a couple of lifetimes, which we'll describe in the next lecture, there was a search for proteins that only have a single tryptophan residue in them. And the search was taken a little further to look for proteins that really looked like only the 1lb emission, the higher emission. And one of these was this protein azurin from a microorganism. And if you look at the native protein, and if you excite it at 285, it looks like the 1lb spectrum, the structured emission, as opposed to the commonly observed longer wavelength emission. If you denature the azurin, of course you expose the tryptophan residues to the solvent. It shifts to the normal unstructured emission spectra, 350 nanometers. That's the only thing you see at the longer wavelength excitation. If you go to the shorter wavelength excitation, you can see contributions of both tyrosine and tryptophan. But this is shows you how sensitive the tryptophan emission is to even modest contact with water. This next one I think is even more amazing. This could have never been done without the technology for site-directed mutagenesis, but this was another azurin that had a single tryptophan residue in it, and it was noted in the crystal structure, that there were no polar residues around the tryptophan residue. So when you looked at its emission spectrum, it looked like this, the previous one, the structured 1LB emission. And what these researchers did, which I thought was extremely clever, they replaced these two, one at a time. They replaced either the isoleucine residue or the phenylalanine residue, both that are nonpolar, with a single serine residue. The serine, as you might recall, has a hydroxyl group on it, and the placement of the single hydroxyl group around this tryptophan residue shifted it to the unstructured, longer wavelength emission. So it doesn't take much to shift the tryptophan. In proteins, the tryptophan emission vary from fully shielded to fully exposed. Every type can be found if you look at enough proteins. I put a few examples here for you. Buried tryptophan azurin having the structure emission. Ribonuclease T1, which we'll talk about, I think, more in the next lecture, has a buried tryptophan, but it still has polar groups around it, slightly longer wavelength emission wavelength. And just jumping to the most exposed one, glucagon, the emission is that the indole group is just out in the water, and it has a 355 nanometer emission maxima. So the spectra from tryptophan can be all over the place depending on their degree of exposure. Oh darn, I hit the thing again, which made me skip a slide, but I'll tell you what the slide said. The slide said that if you look at indole and different solvents, this is a slide we presented in the first lecture, where we have indole and ethanol, and the smallest amount of ethanol shifts the emission, which is a specific solvent effect, and the larger amounts of ethanol just give it a general solvent effect. I don't know if I can get this edited in or not. I'll give it a try. One thing that has been of interest to the biophysicist has been what is the exposure of these single tryptophan proteins to the aqueous phase. Acrylamide turned out to be a very efficient quencher of indole. It's electron deficient, likes to accept an electron from indole. And here we have 20 proteins or so, and we see that for the most part, except for one or two exceptions, all of the quenching constants are within about a factor of 10. And that says to some people that the tryptophans can be very deeply hidden and not quenched. But I sort of take it to think that the tryptophan is really not a solid little block in a protein, but rather the protein molecules are flexing in the excited state. We did experiments with oxygen quenching, and admittedly, these are multi-tryptophan proteins, but we found that for all the proteins that we looked at, the oxygen quenching constants were within about a factor of 4. And what that meant was that the oxygen was really passing through most of the photons. Both my PhD advisor and myself, we got a lot of grief for that because no one believed that could possibly happen. But it looks like the proteins do have enough flexing to allow small molecules like oxygen pass readily through the globular structures. This is an experiment that you could never do without site-directed mutagenesis. It's just sort of I think, a beautiful experiment. It was known that calmodulin had 4 binding sites for calcium. How there was really no other obvious method where you could independently measure what is the binding constant of each of these sites. The calmodulin had no tryptophan residues in it. Calcium is spectroscopically silent from our point of view. So these investigators inserted tryptophan residues into the calmodulin, one near each of the expected binding sites. And lo and behold, they could discern the different binding constants for each of the tryptophan binding sites by using these calmodulin mutants. And tensor changes were normalized, which made it easier to look at. They even decided that some of the calcium binding sites were cooperative with one another. So I consider this to be a brilliant experiment in protein chemistry. This is really an interesting one. The single strand binding protein that is present in all forms of life. And it's a tetramer. It's wrapped around with DNA. Typically, protein fluorescence tends to be quenched when it's bound to DNA. It binds about 70 nucleotides. But what is the purpose of the single strand binding protein? Well, we have proteins called helicase, which can unwind the DNA. But the problem is when you start unwinding the DNA, you now have single-stranded binded DNA. It can be chewed up by other enzymes in the cells. It can be self-complementary. It could bind up to other oligomers. So nature had to prevent this somehow. So nature invented the single strand binding protein, which as soon as the DNA helicase unwraps the DNA for replication, it gets covered with single strand binding proteins, and these protect the single strand binding proteins from recollapsing and binding to themselves, because they had to stay separate for other processes which were further down the road, like polymerase, where you would have to start replicating the single-stranded strand into a double-stranded strand. Since the proteins are always quenched by binding oligos to them, you could see you could watch this binding just by stop flow measurements, and you can see how rapid it is. Interesting thing about it, even though it looks like it's a random distribution of single-stranded binding proteins, there seem to be discrete sites on the nucleic acid where they like to bind. So there have been some recent single molecule experiments that shows these single-stranded binding proteins jumping along the single-stranded strand rather than moving continuously. What about the correlation of protein with protein fluorescence with 3D structure? This would not have been possible using just fluorescence alone. This was only possible because we already knew something about the X-ray structure of the protein. But knowing something about the X-ray structure of the protein and knowing where you could put tryptophans into this adrenotoxin, a small single chain protein, you could ask, what does the tryptophan emission look like in each of these sites? So the tyrosine and the phenylalanine were replaced. One trip was put on the surface, 82, which I think is this one here. Another one was buried in the protein, which I think is this one here, which is trip 11. And they looked at these proteins, and as would be expected, the tryptophan 82 looked like the typical well-exposed tryptophan residue. The one that was buried, the W11, had a more blue-shifted, shorter wavelength tryptophan residue. In case you're confused by the F11 here, that's replacing the phenylalanine. And the quenching constants agreed with that. That is, the W82 was quenched more than the W11. But even though they're exposed and they're buried, this sort of agrees with what I said previously, that the proteins seem to be permeable to some extent to small molecule quenchers. Another example of this, and I'll do this one rather quickly because this lecture is turning out to be longer than I had hoped. I guess I should have broken this one up. You can correlate the three dimensional structure of antithrombin when some of the other residues were replaced from tryptophans, and to state this very quickly, the tryptophan 49 looks to be right on the surface of the protein, and it's the red shifted emission, which you can see down here. And the tryptophan 225, I think, is the buried one, and it's the most blue shifted of this. I can't help but mentioning the genius of this experiment, because the authors could not replace all of these four residues at the same time because the protein structure was disrupted. So they replaced just one at a time, looked at the spectra of the other proteins that contain the other three tryptophans, and managed to reconstruct the individual emission spectra. So I thought that was quite a tour de force type of experiment to do. This is becoming interesting in drug development, which we'll talk about on the next slide. But that is, you can learn something about protein unfolding by looking at the tryptophan residues as the protein is unfolded. So we have this lactate dehydrogenase here, which was exposed to increasing concentrations of guanidine hydrochloride. And what you can notice is that for tryptophans in different domains, they either denatured very quickly with guanidine, or they were much more restrictive and held together longer with guanidine hydrochloride. And that can tell you something about unfolding of proteins. And as basic as this observation seems to be, it is now widely used in looking for drugs based on monoclonal antibodies. These, if you watch television commercials and and you see advertisements for a lot of new drugs, you'll notice that actually a lot of them, if you look at their generic names, have MAB at the end, which means monoclonal antibodies. And these are being reported in high structures. Latest account over 100 in use. Dupixent is probably the most advertised one, used for asthma and dermatitis and other allergic conditions. And what it does, it turns off the eosinophiles, which are partially responsible for the allergic response. But in order to use these therapeutic antibodies, you don't just swallow them because you'll digest them. You have to ingest them. You have to inject them into yourself. They might sit around your house for a long time before you get around to injecting them. Long-term stability is a problem. So even though I don't have the monoclonal antibody names here, because they're normally a secret, but a denatured antibody will have a different ratio of emission of short wavelength to long wavelength emission. You can use this in a thermal test to see how the antibody unfolds, or perhaps more conclusively, you can use this to look at the long-term storage of proteins. I happen to take one of these, keep it in my fridge for a long period of time, and you see what the drug companies will do. Will test all their monoclonal antibodies over a long period of time, and here are the rates of loss of the monomers, because the proteins over time would tend to aggregate. So if they're going to choose a candidate for a potential drug, they would choose one of the monoclonals that has the lowest amount of aggregate formation after being stored months in the fridge. So what are the final comments about protein fluorescence? I think one is, I think I've showed you, they're highly sensitive to almost any change in conformation, sensitive to substrate binding, sensitive to binding to other proteins or membranes. So even though you might not be able to interpret a spectral change for the protein, you can determine that it's happening when two proteins interact. Some experiments could be done that could not be done any other way, like the calmodulin experiments. One I should have written in here, I think even globular proteins have a reasonable amount of flexibility, so the quenchers can pass through them. And the last point I'd like to make is these detailed structural changes that I showed you could never have been known without knowing the three-dimensional structure, and so they are not revealed, and they were not revealed by our fluorescence experiments, but rather by a comparison with the three-dimensional structures and the where the tryptophans were and what surrounded them. So that's the part of 12A. I thank you for your attention, and we hope to see you next time for 12 part B. Bye for now.